showing first is just some snapshots, some still images of different types of mouse behavior viewed from above versus from below. And the point of this is just to show that even if you're talking about, anthropomorphically defined behavior such as paw biting or licking or scratching, right, grooming, that if you look from above, it's really hard to tell and differentiate between these behaviors. So if you look here over the left-hand column, paw biting versus licking, paw licking, you can see that, like, looking from above, they look like they're doing basically the same thing. But when you look from below, you can see so much more detail, right? You can see exactly where the animal's mouth is and where their paws are. And so, this was the first hint that I got that, okay, I'm on to something, right? So I'd made some devices to look from above and from the sides of the animals. And it wasn't until I put a camera underneath, I was like, okay, now this is the viewpoint that we want. So the next thing we did was to see if I can get this to move forward. So this, I'm now showing a schematic for those of you listening. It's a side view of what this technology looks like, right? So on the top, you have a mouse that's inside of a box. The floor of the box is made of glass, and beneath that glass, you have a camera. In this case, we're using near infrared light to illuminate the animal and to illuminate the paws. Near infrared light is invisible to mammals, so humans and mice cannot see near infrared light. So it gives them the perception, the illusion that they're in the dark. Right. So the animals apparently in the dark, and they're nocturnal, so they feel safe. There's no humans in the room, so they also feel more safe. And we have two different light sources. The first light source is over here on the left, where it says near infrared trans illumination. And this is simply an LED light shining from below the animal through the glass to illuminate their body from below. And then we have a separate set of lights that are shining into the long edge of the glass. So these lights are totally internally reflected. It's a phenomenon called frustrated total internal reflection. And every time a beam of light hits that glass air interface, it creates an evanescent wave. And that evanescent light wave diminishes exponentially as it moves further from the glass. And what that does, it creates a pressure sensitive signal. So as the animal presses their paw down on the glass, the sort of ridges in their skin come more and more in contact with the glass, and you see that that signal between the paw and the glass becomes brighter the more pressure is pushed down. And in this case, because that signal is, on the one hand, you want to see the whole body, which is why we have this transillumination signal. But that transillumination signal can interfere with detection of exactly the precise time when the paw hits the glass. So I'm not sure how clear this will be to your listeners that are not watching the YouTube video, but if you imagine you're have a flashlight looking through a piece of glass, and on top of that glass, a mouse is walking around. Well, if every time the mouse puts its paw down, the paw creates a little light signal, and you're also shining light from below, those two signals are going to kind of cancel each other out, and you're going to be able, and you're not going to have as clear a distinction as you would if you didn't have the flashlight on. And so there are a couple of ways that we've used to mitigate this issue and correct for it. And one is to use simply two different wavelengths of near infrared light with and use two cameras, right, with a bandpass filter on each one, so that the wavelength of light you're illuminating the animal is different than the wavelength of light that you're illuminating the paws. And the other way we've done this is to alternate the illuminations on different video frames. So one video frame, you look at the paws, and the next video frame, you look at the body, and both work equally well.
But I guess the punchline here is, you've removed humans from the environment. You're doing this in the dark in a situation where the animal's sort of maximally comfortable and able to go about its business unfearfully. And two, you've used cameras and infrared light in a clever way such that every time that a mouse steps on its paw, you can literally see how much pressure that's being put on.
Yeah, exactly. And I've got some videos here that really are videos worth 1,000 words. So on the left is the paw image, on the right is the mouse, and then I'm bringing them together here and kind of overlaying them so that you can see what that looks like. And this is just showing a mouse sort of just, he's scratching, kind of walking around a little bit, but with the signal from the paws, we've color coded it so that it's like a heat map, and that way you can sort of intuitively see what the mouse is, how much pressure is both within each paw and what the difference in the pressure is between the two paws. Yeah, no,
I mean, it makes perfect sense. Like I'm looking at this mouse, I can tell just by looking at it from below that it's standing back on its hind paws and not on its front paws. But then the heat map makes it clear what the computer is going to see, which is the pressure is on those hind paws and it's not on those front paws so much.
Right, exactly. And so I've got a couple more videos here. This next video is showing sort of the workflow. So, it's not enough to simply, show the mouse walking around and detecting limp, which I'll show you in a moment. We really wanted to be able to automate this and let machine learning algorithms tell us, what's going on. And so in the workflow there is that we basically, used these machine learning or machine vision algorithms rather to identify the four paws and the tail and the snout of the animal, and then on each video frame to orient the animal so that it's always facing the same direction. And this makes it much easier for subsequent analysis of each video frame, because you always know that the left paws are going to be on the left side of the image and the right paws are going to be on the right. And it's not going to change when the animal turns around and goes the other direction. And then the next video here is showing, the unaligned frames. And this is where we've used the algorithm to look at each of the paws, and the yellow here is identifying the left hind paw of the animal, and the blue is identifying the right hind paw of the animal. And we can also use the same algorithms to identify each of the other paws. And I want to point out here, we're using a lot of some off-the-shelf machine learning algorithms. Deep Lab Cut is a toolkit that we find really helpful to work on this. And we actually have a paper that's under review right now, so I don't want to preempt where it's going to be published, but we do expect to publish this in the next couple of months, and all of these algorithms and the source code there will be made publicly available. So
interesting. Yeah. So now you've got a way to it's really an objective way to measure pain in a more naturalistic setting.
Yes. And, what we find is that we can, in doing this, not only do we see that it's really clear when an animal is in pain versus when they're not in pain, in particular if the pain is, in one of the hind limbs, but we also see that we can reverse that pain with a pain drug at a much lower dose than necessary to reverse reflexive withdrawal. And so it's a much more sensitive assay, and it's backward. It backwards translates all existing analgesics work really well, and it's our hope that it will also be quite predictive of future analgesics, and that we'll be better able to predict. And the last thing I'll mention that I think is I'm really excited about is that whereas typically we have to use somewhere between 10 and 12 animals typically per group to get statistical significance in a pain assay, with this approach and the machine learning analysis of these data, we can do the same thing with 3 or 4 animals. And so we're cutting, the number of animals needed to be used pretty dramatically. And additionally, we can actually do high throughput screening of behavioral phenotypes of pain and analgesia in mice very efficiently.
Mm-hmm. I would love to start talking about some of the different kinds of pain drugs that are out there and how they're actually interfacing with the nervous system. So if we go off of the screen share, I think a good place to start, perhaps. Oh no, do you have a video that's good for this?
Well, yeah, actually, let me show one last video here of the pain.
Okay, yeah.
This goes with what I was just saying. So this is a mouse pain model on the left, and you can see that his left hind paw is not as bright as the right. It's the one he's scratching there with, and and then when he the same mouse, after we give him an analgesic, and you see that those hind paws are now equal, right? So on the left, he's really favoring one paw over the other, and on the right, he's no longer doing that. So the thought here is that we can use some of these same tools that we can use to detect pain readouts in rodents, that we can use some of these same tools to differentiate between some of the different phenotypes behaviorally and experientially of broadly speaking psychedelics. And I'm using that term loosely to include entheogens and dissociatives, which I think we can likely differentiate using this black box detection technology.
I see. So one potential application here is, so again, this comes back to the problem of, we like to, in general, in research, people like to do work in animal models first and then pursue promising avenues that come from that in humans. So just like you can't ask a mouse if it's in pain and you have to come up with clever ways to assess the pain, you also can't ask a mouse if it's tripping on acid. You have to come up with some other way of doing that. Do you want to mention how that works today and how you might tie into that?
Yeah. So, right now, there's the psychedelic space is an interesting space because there's been a lot of anecdotal and really well-developed experiential literature on how people have experienced and used plant-based psychedelics, not just in modern times, but for quite a long time. And there are benefits and I'm certain a lot of uses for the naturally occurring psychedelics, but I think there are also a lot of potential use cases that could be developed using synthetics that are derived from these same compounds, and we can fine tune them to different indications and use cases and maybe find ways to fit them better into our current healthcare system. The difficulty there is that, or I should say a difficulty is that when you develop any new drug that's a new molecule that does not exist in nature, before you can test it in humans, you do have to test it in animals, both for efficacy and safety. Safety is something else that we're doing with the black box technology, but on the efficacy side, psychedelics, there's really just one readout behaviorally that is used to detect sort of the hallucinatory type properties of a psychedelic, and that is the head twitch in mice. And this is a rapid rotational movement that mice exhibit from all of the classical psychedelics, but it's a very infrequent behavior and it's very difficult to detect. And so you end up having to literally have people watching videos of mice for hours on end and writing down, a handful of head twitches that happen here and there. And the problem with this is that while it is quite sensitive to psychedelics, it's infrequent enough that it means that a lot of animals have to be used. It's expensive to do, and it takes a lot of time. So the first thing that we can do with the black box technology is that we can automate this, right? So instead of people having to watch this, we can automate it and just right out of the gate, detect these head twitches automatically. But beyond that, I'm convinced that we're going to see quite a few behavioral differences using some unsupervised machine learning approaches, where we will be able to differentiate not only between is this a psychedelic or creating hallucination-like effects or not, but even to sort of to really tease out, is this a compound that has entheogenic or empathogenic type properties? Is it anxiolytic, right? Mm-hmm. So a few of your previous guests have talked about, the psychedelics tend to act on the serotonin H2A receptor, which produces hallucinations, but also potentially a lot of the therapeutic beneficial aspects of psychedelics. But you also have the HT2B receptor. And so the HT2A, in addition to these, causes fear, right? Which is why, if you take a really high dose of LSD, people report, that they can have some really fearful experiences. Whereas the HT2B receptor, which is activated by compounds like MDMA, 6-APB, some other compounds, can actually be anxiolytic. And so we've got some readouts. I'm happy to actually show you a little video clip of how we can read out in real time using different measures that I showed you earlier, the present state of anxiety in a rodent using the same technology. So if you want, I'll show you another little video this of this. So let me go to this next slide. So anxiety in rats. So if you look at on the left here, we've got a rat. This is the same rat. So for those of you listening, there's a video of a rat walking around in a chamber, and his paws are illuminated using this touchpad technology, the Blackbox technology. And if you look at the contact points of the rat, it looks like he's standing on the pads of his feet. Mm-hmm. Instead of sort of the outline of a footprint, you see these points of light that are sort of round points of light, like at the balls of his feet, right? And so that's what all the rats and mice for you're showing this so that we show that it works in both rats and mice, but see the same thing in mice. And that's what all the rats look like after they've just been handled. But if you leave the rat in the room and you leave the room, you see an entirely different footprint. So this is the same rat 10 minutes later. So I've left the room, just left the rat to hang out there, and they get flat-footed. And if you think about it sort of intuitively, it makes sense. It's almost like I think it's a vigilance readout, right? Mm-hmm. So, if a rat is scared and sort of wanting to be ready to run, they're kind of up on their tiptoes, just like a human sprinter would get on their toes to start a race, right? Mm-hmm. And when they're calmed down, they get flat-footed. And so we have this anxiety-related footprint, and that's dramatically different from the calm or the anxiolytic footprint. And we see this. It's not just after human handling. You can even if you go to the door of the room where these animals are housed and you just jiggle the doorbell, it'll immediately go from this flat-footed appearance to on the tiptoes again.
I mean, this makes a lot of sense. And I guess, the idea here overall is, using these objective readouts, you can not only do behavioral assessments faster and more cheaply, you can actually just detect things that you wouldn't have been able to detect before. And this isn't limited to pain. It's in theory anything.
Right. No, exactly. And I'll, just to mention a similar thing that, because I, really have this interest in the psychedelic space. So this is actually a video of, it's actually a cropped video that I showed earlier. And this is just showing a flinch and lick response. So I'll play it again. So if you watch what this mouse does, so let me see here. I got to think. It's his left hind paw is injured. He has an inflammation model. So his left hind paw, which is a little less bright, is presumably in pain, and he kind of flinches it and then licks it, right? And so I'll stop the video here right at the point. So right here, he flinches, he like shakes it, and then right after he shakes it, he licks it. And this is a very common sequence, right? And so if you apply a heat stimulus to the foot of a mouse, they do the same thing. They flinch it and then they lick it. But with dissociative drugs like ketamine and other compounds, they will flinch,
but not lick.
You see that they have this pain phenotype, behavioral phenotype, but they don't lick it. They don't attend to it, right? It's like they're aware that it hurts, but it's like not bothersome, maybe. I'm, anthropomorphizing here, but point being that this is another behavior that we can automate, right? Mm-hmm. So, and you can imagine when you start stacking these behaviors on top of one another, it makes it quite easy to tell for a novel compound, is this, if you're a drug, psychedelics development company, which is a lot of activity in this space, and you've got 20 new compounds, and you think they're likely from some of the cell-based assays and other approaches to be have psychedelic-like properties, but is it more like MDMA? Is it more like an LSD or psilocybin, or is it even potentially a dissociative compound, right? Mm-hmm. And so we can behaviorally really distinguish between all of those, and that's sort of the next thing that we're doing there at Blackbox commercially, is that we are in the process right now of setting up a contract research organization in the Dallas-Fort Worth area to do high throughput screenings for third parties. So we're not doing any of our own drug development work. We're simply setting this up as an assay to provide service to the psychedelics drug development space for anyone that's developing compounds and really wanting to tease out, what kind of compounds do they have prior to entering into humans.
I see. So it sounds to me like there's two things going on that will synergize in terms of what you're doing with this.
Right.
One, you could just have someone, let's just say you had a research lab, whether it was academic or private, you were interested in screening compounds to see if they might be hallucinogenic. Today, the way that people do that is they have a researcher spend probably hours and hours looking for this head twitch response in video. Right. So you guys can just automatically detect that so that they can do that type of screening faster.
Right.
The second thing that it sounds like you're doing is you could, if you gave different groups of animals different drugs, by videotaping them and using machine visions in the way that you've shown us, you could create a kind of behavioral fingerprint for each drug. Right. In other words, animals, when they get MDMA, they tend to do these types of behaviors, but not these ones. Those that get LSD, there might be some overlap, but it's different. Even something like psilocybin versus LSD might have small differences between them. And then you could take a completely novel drug, say, and just compare it to all of those known drugs with known effects in rodents and known subjective effects in humans. And in theory, that should be able to give you quite a bit of predictive power for what a novel drug will actually feel like.
Yeah, we hope so. And I think that will be the case.